Hello my students, how are you all? Welcome back to my EduSpace. Hope you all are doing and learning well. If you like my videos, then don't forget to subscribe my channel and you can share to your friends as well. Let's talk about today's video. It is related with the cells of immune system. In today's video, we are taking lymphocytes. Three types of lymphocytes are present in our body, that is B-lymphocytes, T-lymphocytes and natural killer cells, or we can say NK cells. B-lymphocytes and T-lymphocytes are participated in humoral and cell-mediated immunity respectively, whereas NK cells are participated in providing innate immunity. But including all three types means the video gonna be very large. So today I will took only two classes of lymphocytes, that is B and T lymphocytes, and in next video we will take natural killer cells. So without further ado, let's get started. Let's start our topic with the introduction. So lymphocytes are having antigen receptors on their membrane. So these cells carry receptors on their cell membrane and plays a central role in adaptive immunity. Okay, so suppose this is a lymphocyte cell. They are having characteristic features of bearing a receptors. This receptors, when it interacts with the antigen, it stimulates or activates the cell, which ultimately takes part in immune responses. So it becomes activated. They are having the properties like diversity, specificity and memory. So the diversity indicates there are millions of lymphocytes that are characterized by the receptors that are present on their membrane. In the term of specificity, it means they are very specific. Specificity in case of types of antigens. For a particular type of antigen, there is a particular cell receptors on the membrane of these lymphocytes. So specificity, suppose we are having two types of B lymphocytes and they both are different from each other because they express different types of receptors. This particular is for antigen 1 and this particular receptor is for antigen 2. So they are very particular or specific in their action. And last one is memory. Lymphocytes are differentiated either into effector cell or memory cell. So as soon as they interact with the antigen, they are differentiated either into the effector cell, okay, or a memory cell. They have long lifespan as compared to effector cell. So this memory cell is lifelong or having long lifespan as compared to this effector cell and provide heightened and more rapid immune response if the same pathogen affect the body again. So 20 to 40% of WBC are lymphocytes and 99% is present in lymph. So 20 to 40% of WBCs are our lymphocytes and in the lymph their number is 99%. Their number in our body is approximately 10 to the power 12 or trillions in number. Lymphocytes circulates in the blood and lymph and can migrate into tissue spaces and lymphoid organs. Now, types of lymphocytes. So before we are taking types of lymphocytes, let's recall ourselves that how these lymphocytes are formed and from which kind of progenitor cells these lymphocytes are formed. As I already told you in our previous class that there is a hematopoietic stem cells, okay, which having the capacity of self renewal. This particular hematopoietic stem cell can be differentiated into two types of progenitor. The first one is myeloid, okay, and the second one is lymphoid. So this lymphoid progenitor is responsible for the formation of the lymphocytes, okay. So from this lymphoid progenitor cells, three types of cells are formed. The first one is B lymphocyte, T lymphocytes and NK cell. So let's talk about types of lymphocytes. Broadly, we classified lymphocytes into two categories, okay. The first one is B lymphocytes and another one is T lymphocytes. But natural killer cells or NK cells comes under this category also. So their lymphocytes are classified into three categories. The first one is B lymphocytes, the second one is T lymphocytes, and the third one is natural killer or NK cells. This is the picture or diagrammatic line diagram of the lymphocytes. So first take this B lymphocytes. You can see on their surface membrane, there are Y-shaped structures are present. These Y-shaped structures are immunoglobulins. These are immunoglobulins, okay? Or we can say antibodies. And in the T lymphocytes, you can see there are CD3 receptors, T cell receptors, and this one is our, what is this? This is CD4 or CD8. The CD4 is present in TH lymphocytes and the CD8 is present in the cytotoxic T lymphocytes. Now the NK cell. This NK cell, you can see there is no types of receptors or surface marker is present on its surface, okay. So this is our NK cell. So let's learn something about the cell cycle of lymphocytes. Before antigen activation, okay, the B and T look very similar to each other and they cannot be distinguished. This time they are known as naive or unprimed. Here I am taking the example of B lymphocytes, but both types of lymphocytes are possessing the same kind of cell cycle. This time they are very small in structure, okay. So they are called naive or unprimed because they are not introduced with any kind of antigen. In activated state, they are getting interacted with the antigen. So in the first, when they are naive or unprimed, they remain at the GO phase of the cell cycle. The GO means the resting phase where the cell resides until it get activated, okay. So in next they comes to the G1 phase or gene activation phase where they are introduced with the kind of antigens. After interaction with antigen, they progress from GO to G1 and S to G2 and M phase, okay. As cell cycle proceeds, lymphocytes enlarges. It becomes enlarges and now this is called as lymphoblast. This lymphoblast is formed during the S phase or DNA synthesis phase. Lymphoblast proliferates and differentiates into effector cells or memory cells. So during S phase, the lymphoblast is formed and from the S phase, they go to the G2 phase and then M phase. M phase means here is the proliferation of the lymphocytes takes place and they multiplies. and after that they differentiated either into effector cell or memory cell. The effector cell, this one, it is also called as plasma cell, and it helpful in eliminating antigen because it is the production house of antibodies. They produce large number of antibodies which are specific to that particular antigen. And in T lymphocytes, there are two types of effector cells produced. The TH cell, which secretes cytokine, okay, and TC or cytotoxic T lymphocytes, that is known as CTL, that are antigen activated mature lymphocytes in case of T cell. The memory cell, it provides lifelong immunity to many pathogens. You can see one thing, that the memory cell is quite similar to that naive or unprimed cells or immature cells, but they are different from each other. How they are different? They can be distinguished by the presence of surface markers. You can see the surface marker here and here. They are quite different. How they are different? Because this is unprimed and this one is specific, as I showed in the green color. This particular is specific, or we can say the complementary to that particular antigen. So this one is specific to that pathogen that has activated the lymphocytes during G1 phase of the cell cycle. So let's revise again the cell cycle of lymphocytes to make you understand better. So first, it is very difficult to distinguish the B and T lymphocytes. So the first one, which is at the GO phase, these lymphocytes are known as naive or unprimed because they are not introduced with any kind of antigen. So as soon as they are introduced with the type of antigen, the gene got activated, okay, and they goes to the G1 phase. And during the G1, during the synthesis, DNA synthesis, they form the lymphoblast, okay. This lymphoblast during M phase or mitotic phase become differentiated into clones of the lymphocytes, and then they get differentiated into the memory cell or the effector cell. In the case of B lymphocytes, the effector cells is known as plasma cell, which is the production house of antibodies. They produced so many antibodies which are helpful in eliminating the antigens. But in case of T cells, they are differentiated into either helper T cell or the cytotoxic T lymphocytes or CTL cells. In both cases, the memory cell is formed, and the memory cell is specific to that particular antigen which is introduced during G1 or gene activation during GO phase, okay. And it is quite similar that both the lymphocytes that one is present in the memory cell, okay, and that one that is at the naive or unprimed GO phase. They both are quite similar in structures or in morphology, but they are different. They are different in their surface markers. Their surface markers, or we can say the antibodies that are present on the surface of the B cell, which is at the GO phase, is different to that of the memory cell. In memory cell, the antibodies are specific to that particular antigen which is introduced at the GO phase and which caused the activation of the B lymphocytes. Okay. Now, B lymphocytes. It derives its letter B from the site of maturation. Initially, the B was taken from the bursa of Fabricius that is present in birds, but now it's apt that it is for the bone marrow, which is the maturation sites of B lymphocytes. They can be found in primary lymphoid tissues like bone marrow or in secondary lymphoid tissues like spleen or lymph nodes. They distinguished from others as they display membrane-bound immunoglobulins or antibodies as receptors for antigens. So you can see this is the B lymphocytes, but this is naive B lymphocytes. Okay, and what does it possess? It possesses these surface markers, and these surface markers are known as immunoglobulins. These are membrane bound immunoglobulins, or we can say the receptors for the antigens. How they work? These cells produce antibodies to fight against bacteria and viruses. These antibodies, or we can say immunoglobulins, are Y-shaped structures. These are Y-shaped structures, or we can say Y-shaped proteins which are specific to every pathogens. Okay, they are very specific to every pathogen and lock onto the surface of an infected cell and mark them for the destructions by other immune cells like phagocytes cells or macrophages. So antibodies on the membrane of single B cell has identical binding sites for antigens. So you can see there are millions of B lymphocytes or B cells are present, but they are different from each other by the presence of these special or specific surface markers. These surface markers or binding sites of antigens are different for each types of B cells. As antigen encounters B cell, it differentiated into effector cell and memory cell. So suppose this is our naive B cell which is not right now introduced with any kind of antigen. As soon as the antigen comes, it interact with the receptors of the B cell, or we can say the immunoglobulins that is present on the B cell. Okay, it differentiated into the plasma cell, or we can say the effector cell and the memory cell. This plasma cell is having lots of they are having lots of endoplasmic reticulum and Golgi apparatus, because they are in increasing demand of production of antibodies. So these antibodies are basically protein structures which are helpful in eliminating the pathogens. So how these antibodies works? They basically they lock onto the surface of an infected cell. Suppose this is the infected cell, they lock the pathogen. Okay, they lock the pathogen and then mark them for destruction by other immune cells like phagocytic cell. Then this, as soon as it locked up the antigen, it comes or get interacted with the phagocytic cell, which ultimately causes destruction of this particular type of pathogen or antigen. So let's talk about T lymphocytes. As B cells are responsible for antibody mediated immunity or humoral immunity due to antibodies, whereas T cells are responsible for cell mediated immunity. The T lymphocytes, or we can say T cells, it derives its letter T from its sites of maturation, that is thymus. Okay, so as the site of maturation is thymus, so the initial letter is taken from this thymus. Functionally, T cells have solid role in adaptive immunity. It has a role in adaptive immunity. It does not take part in innate immunity. They kill infected host cell, that is cytolysis, can activate other immune cells by producing cytokines and also regulating or suppress the immune responses. The T cells are having central role in generating immune responses against viruses. So what are the roles of T lymphocytes? They kill infected cells, okay, and they can activate other immune cells by producing cytokines, and also they can regulate or suppress the immune responses. During maturation, it expresses T cell receptors on its membrane. So, suppose this is our T lymphocytes, okay, and it's having TCR. This is known as TCR, which means T cell receptor. This particular receptor is able to recognize the antigen, but that antigen should be bind to the MHC molecule. Okay, so we will discuss later what is MHC molecule and how many classes are for MHC molecules. So like B cells, which can like B cells in which the surface markers or the immunoglobulins can detect the antigen, which is not essentially to bind to the any kind of MHC molecules. But in the case of T lymphocytes or T cell, it can only recognize or bind to the antigen once it binds to the MHC molecules. Okay, so just read it out. Unlike B cells, T cell receptors recognize antigen only when it binds to the cell membrane proteins called major histocompatibility complex or MHC molecules. So this particular antigen can be recognized by this TCR if it binds to another class of molecules, that is MHC molecule, which is present in a kind of immune cells, which may be macrophages or nucleated, any kind of nucleated cells. Now let's do some study about how the T cells get bind to the MHC molecule, which is presented by antigen presenting cell. So there are basically two major types of MHC molecules. The first one is class 1 MHC molecule and the second one is class 2 MHC molecule. The MHC class 1 expressed by all nucleated cells. So class 1 MHC molecule is expressed by all the cells which contain nucleus, whereas class 2 MHC molecule is expressed only by some kind of special cells. These are known as antigen presenting cells. They may be B cells or macrophages. Okay, so suppose this is our T cell. This is T cell. Okay, so this particular region is known as, this particular surface marker is known as TCR. This is T cell receptor, which is going to be interact with the antigen which priorly bind to the MHC molecule. This particular is, these two are CD molecules. That means cluster of differentiations. Okay, so this one is CD3, which is present as a surface marker on the T cell, and this one is CD. This can be either CD4 or CD8. There is much difference between these two CD4 molecules, CD molecules, because if the CD4 is present at the surface, then this will differentiate it into helper T cell, and if this CD is 8, then it gonna be cytotoxic T cell. Okay, so suppose this is our T cell and this gonna be bind to the antigen which already bind to the MHC molecule with present by antigen presenting cell. So this cell may be macrophages or any phagocytic cell. Once they get interact, this particular T cell will get either differentiated into helper T cell if this CD molecule is CD4. Okay, if this is CD4, the cluster of differentiation is CD4 molecules, and it can be differentiated into cytotoxic T cell if this CD molecule is CD8. Okay, this is CD8. We are representing CD8 by a single line, whereas CD4 by double line. Okay, and this T lymphocytes can also be differentiated into memory T cell. So basically, what are these macrophages? Basically, these are phagocytic cells. Suppose this is antigen or any kind of foreign invaders, and this gonna be engulfed by macrophages. Okay, so once they engulf through the digestive enzymes, they are breaking down into simpler molecules or peptide fragments, and this particular peptide fragments is presented by the MHC molecule that is present on the surface. Okay, then this particular MHC molecules is expressed on the surface with a peptide fragment of that particular antigen. So this is peptide fragment of antigen, this one. Okay, so let's talk about the TH cell or helper T cell. So this is our naive TH cell, which can be said that this is unprimed or before introduction to antigen. So once the TCR is introduced with the antigen that is bind with the MHC molecules. This MHC molecule is MHC class second. Okay. Therefore, this antigen presenting cell is macrophage. This is macrophage or any kind of phagocytic cell. So this macrophage, which breaks down this antigen into smaller peptide fragments and which ultimately expressed by MHC class second molecules. Then only this antigen MHC complex is recognized by this TCR, T cell receptors. This one is our CD4, okay, which ultimately leads to the formation of helper T cell. Now, as soon as this interaction takes place, the TH cell get activated and can be differentiated into TH1 cell or TH2 cell. The TH1 cell will release TH1 cell and TH2 cells can be differentiated by the types of cytokines they produce. The TH1 cell produce IF-1 gamma, whereas the TH2 cell will produce the cytokines that is interleukin 4 and interleukin 10, IL-4 and IL-10. So the T helper cell, it displays CD4. As I just told you, it has the surface marker of CD4. After activation with antigen MHC complex, it differentiates into effector cells and memory cells. As I just told you that after the activation, when it combined with this complex of MHC and antigen, it activates and can be differentiated into effector cells and memory cells. And these effector cells can be T helper cell type 1 or T helper cell type 2. It helps in activation of B cells, macrophages, and other cells that are involved in immune responses. So as they are activated, they are ready to form cytokines, and these cytokines are responsible for the activation of B cells, macrophages, and other cells that are involved in immune responses. Now, the another subpopulation of T cell is the cytotoxic T cell. It is just same like the TH cell, but in this case, the CD instead of CD4, there is a CD8 molecules, okay, and this one is CD3, which is present in both TH cell as well as TC cell, and this is our TCR, T cell receptor. So the cytotoxic T cell displays CD4, and after activation with antigen MHC complex, this is antigen MHC complex, and this one is MHC class 1 molecule, MHC class 1 molecule, okay. The cytotoxic T lymphocytes or memory cell generated. So it differentiated into cytotoxic T cell or memory cell. This cytotoxic T cell have the ability to eliminate antigen which is complexed with class 1 MHC molecules like tumor cells, virus infected cells. So this particular cytotoxic T cell have the ability to eliminate the antigens which is present with class 1 MHC molecules, or it can kill the tumor cell as well as viral infected cell. Now there is another type of subpopulation of T cells which is known as T regulator cell. This cell express both CD4 and CD25 on their membrane. So this is CD4 and CD25. Okay. What is the function of this T regulatory cell? As its name indicates, it regulates the immune responses. So basically it suppress or regulate the immune responses, and they are negative regulators of immune responses. So if the cell is activated and producing cytokines again and again, then T regulatory cells or T reg cells used to give we can say it can suppress or give negative regulators to that particular activated cell to stop the production of cytokines as soon as all the antigen are eliminated from the body. So these are basically regulatory cells. So how they form? Basically they also form from the naive TH cell. Why? Because they both naive TH cell contain the CD4 molecule. So we just learned about how this TH cell will differentiate into TH1 and TH2, and this TH cell is also differentiated into T regulatory cell, but in the presence of TGF beta molecules. Okay. So with the presence of TGF beta, when this naive TH cell get interacted with the complex of antigen and MHC, then it will differentiate it into T regulatory cell, and it starts producing the same TGF beta cytokines. Now just take a quick recall how the naive CD4 T cell differentiated into TH1, TH2 or T regulatory cells. So this is our naive CD4 T cell. Because why it is CD4 T cell? Because it's having the surface marker CD4 on it. Okay. So as its name indicates, the CD4, this CD4 can be differentiated into helper T cells. because helper T cell contain this CD4 molecules, but we also know that this CD4 can also differentiated into T regulatory cell because it also contain the CD4 surface molecules. So if this naive CD4 T cell, or we can say TH cell, get interacted with the complex of antigen and MHC class second molecules, then they can be differentiated into TH1, TH2 or Treg cell. So how they stimulate to form the TH1? Basically, in the presence of interleukin 12, they can differentiate it into TH1 cell and they start producing cytokines named as IFN gamma. Okay. And just like that, if this in the presence of IL-4 interleukin 4, this CD4 T cell get differentiated into TH2 cell or helper type 2 cell, and this will start producing IL-4 and IL-10. Whereas in the presence of TGF beta molecules, this naive CD4 T cells once get complexed with the antigen MHC class second molecule complex, they get differentiated into Treg cell and start producing TGF beta. Sorry, I just forget to tell you about what are these antigen presenting cells. So just I'm taking you 1 minute to tell about the antigen presenting cells. Basically, those immune cells that boost immune responses by expressing antigens on its surface to other cells of immune system, especially T cells. These are antigen presenting cells. Basically, they boost up the immune cells by expressing their antigen on the surfaces, okay, which ultimately get detected by another type of immune cells, that is especially these are T cells. They are also known as phagocytes. These antigen presenting cells may be macrophages. Okay, they may be macrophages, dendritic cells, as well as B cells. That's all for today, and if you have any query, please text me below and share your suggestions about the topics you want me to cover in upcoming videos. Thanks for watching.